But a transgene cannot function on its own. It requires another piece of DNA called a promoter to tell it when to switch on and off. So scientists always attach a promoter gene onto the transgene before inserting it into the agrobacterium. The promoter can even tell the transgene where exactly in the plant to switch on, which gives the genetic engineer a high degree of control. Genetic engineers usually tag on another piece of DNA called a marker. This is a gene that does something that is easy to spot. If the marker is present, one can be sure that the transgene is there as well. This entire assembly is called a DNA construct. The new gene is now ready for transfer into the target plant. This is done by soaking bits of ordinary plant tissue in a solution containing the transformed agrobacteria. The DNA construct has now moved across and invaded the plant cells. These bits of transformed plant tissue are then placed into culture plates, where they are nurtured into plant embryos, exploiting the ability of plants to clone themselves through asexual reproduction. These embryos are then screened for the marker DNA, which in this case is a gene that protects the embryos from a toxin in the culture medium. Any embryo that survives has the marker and therefore the transgene itself. Once the selected plants are fully grown, they revert back to sexual reproduction and seed formation. These seeds and those of future generations will now contain the new DNA construct.